Good afternoon.
Good afternoon, everybody.
Hello, world.
Hello.
Hello, hello.
So I hear we're looking for a flat, slabby rock around here.
And Kira says she'd like to see if it will withstand a pressure test, if you can break it. If it does, we don't want it.
Roger. So I can kind of jump up to what center screen there. Looks like there's some smaller bits that we might want.
Yeah, sounds like a plan.
Roger that.
Well, hello, everybody. It's afternoon here in California, and we've had a shift change. We've got on the Nautilus crew, Jonathan on video, Jake piloting the Argus, Jessica piloting the Hercules, and Renee doing nav for us. And at the back, we have a science team from Scripps Oceanography in San Diego. Paul Jensen leading with myself, Greg Rouse, and then Caitlin Kramer and Kyla Pearson, who are both graduate students in our labs. And this afternoon we're on the South Patton Escarpment. And presently we're at 670 meters. We're going up probably another 100 meters or so. We'll be collecting rocks, looking for various kinds of incrustations. We're hoping these rocks will have what's called phosphorites, and also animals associated with these crusts, as well as samples that will give us interesting microbiology.
Just for context, I have on the HYPACK screen the bathy with the contours. So we've come a bit up that first slope, and now we're on a flatter section. And then there'll be another steeper slope, and we'll be at the top of this ridge feature right here. So we'll reach the peak and then kind of ride along the ridge for a bit. And I'll bring up the backscatter. It should be pretty bright. The entire, we're on kind of a harder section here, and slope map.
Great. What depth are we looking at when we get to the top of that?
Jonathan, please push on in.
Let's see.
Thank you.
Should be 560 meters.
Okay, so we've got about 100 meters of rise ahead of us.
Mm-hmm.
So Greg, Paul, here's a kind of a flat rock here. Is this something that Kira would be interested in? I can pick it up and show you if you'd like.
Yeah, let's have a look how flat and flange-like it is. She said she didn't really like the lumpy one so much, but she can, well, she's probably getting lunch, but we'll see if she can chime in.
Raj.
And Jess, I think you got the message from Lisa that she wants to try to keep anything on the top of the rock, any animals remaining on the top of the rock. So she didn't like the idea of turning it upside down if possible.
Roger that. Not that much spinning.
Which, yeah.
Actually, adjust my grip force. I'll just do a little rotation so we can see what this could possibly look like.
And if you could squeeze it, if it withstands your maximum pressure, I think
Roger that.
It'll be, looks like a pretty good rock to me.
Do a little more squeezing action here. This is my full grip force here. Oops, sorry. Yeah, so it doesn't look like it's breaking. Still interested in it?
Yes, please. Let's.
Roger that.
We're going to aim for one of the forward boxes on the starboard bio box, please.
Roger. Full wide, please.
Caitlin was speaking.
Okay, thanks. Just watch the mic.
And then the forward boxes you said?
Yeah. Can we go for the outboard forward box?
Roger that. So there is a floaty thing in there. What was that?
Ooh. Oh, no, that's probably a
It's
Worm.
worm from one of the earlier rocks.
Yeah, it was a spaghetti worm.
Roger
Uh-oh.
It must have been already swimming up.
Pretty hard to keep hold of those things. When they want to leave, they'll leave.
that. Escape artist there.
Beautiful.
All right, forward, outermost, in we go.
Great. So this is going to be sample NA124-132, and this is going to be rock number 4.
132.
Beautiful. Thank you.
Sure thing. Anything else around here that you guys are interested in?
Not for me, up to you, Greg.
No, thanks. I think we could carry on along the track, see if we can find some corals.
Roger that.
That'll be bearing 265.
265, Rog.
265.
So it looks like they've done 2 transects in the first watch, so we'll be trying to do 3 more at some point during this 4-hour watch.
Where did that rock go?
Bridge nav.
All green. Thank you.
Step 50 meters, bearing 265, speed 0.3. How'd we do for sediment cores on the first watch?
We actually filled every
Fantastic.
single sediment core that we
Oh, that's
have on
great.
Herc.
It's going to be a busy night.
Nice. And do you have separate ways of processing depending on who they're going to, or is there a general process?
Yeah.
Yeah.
So we're more interested in the bacteria that live in the sediments, and we're basically sectioning them in 1 centimeter sections starting from the surface, and then subdividing those sections for different purposes to do some molecular work and do some cultivation work, and what we call metabolomics, which is something that we've started doing more now directly on sediments, where we look to see if we can detect some of the molecules that we're interested in finding directly in the sediments, and then link them back to the bacteria that might be producing them. So that's sort of an inverted approach to traditional microbial natural product discovery, where usually usually you start with a microbe and grow it in the lab and try to find the product. And so now the techniques have become sensitive enough that we can detect very small amounts of these compounds directly in ocean sediments. And this is kind of an exciting new avenue for us because we're actually trying to see if we can find new molecules right in the environment. And we can identify them that way and make some predictions about who might make them and what sort of genes would be involved in making them, then we can sequence the DNA in that sediment sample and see if we can find the producing organism. So it's really inverting the paradigm of microbial natural product discovery. Instead of going from microbe to compound, we're trying to go from compound back to microbe. And that allows us to potentially find things that are made in nature that aren't produced in a laboratory when you grow these bacteria. So that's one of the big limitations, right? We can isolate bacteria from these sediments and grow them in the lab, but obviously the conditions are very different, and they may not be very interested in producing some of the molecules that they make in the bottom of the ocean when we grow them in the laboratory. And so if we can find the molecules right here, this is basically our petri dish, right? This is our big bioreactor at the bottom of the ocean. And we can capture a snapshot of all of the different molecules that are being produced in these communities and how different organisms might be using them to compete and to acquire nutrients and for other reasons. It's a pretty complex snapshot, but we're now beginning to be able to tease it apart and start to recognize individual molecules. And in some cases, we've been able to make some predictions about which microbes are actually producing those molecules. And so this is an exciting new avenue of research for us. So some of the samples we're saving to do these metabolomic analyses as well. So the other, Lisa's group is more interested in the invertebrates that live in these sediments, what she's calling the microfauna and the myofauna. She may even call some of these macrofauna, I'm not sure, but they'll basically filter the sediments through various sieve sizes and collect various groups of animals and then count them and identify them just to get a census of who's there. So what do you think, Greg? Should we do a transect while we're cruising along? Knock one of those out. How do we know when the last one was done?
It's pretty recently.
Was it recent?
55 meters ago.
Okay, so maybe we should wait a little bit.
In distance time.
Yeah, let's get up to 600 meters and then do one.
So one of the things I'm really hoping we can collect are some additional octocorals, in particular some of the branching whips and gorgonian types of soft corals. So I think in some ways the pilot's eyes are better attuned to some of these things, and you have to see them a little bit ahead of time, so there's a chance for the ship to slow down and stop and us being able to make a collection. So I don't know, maybe we'll make some prizes to see who can find
Keep our eyes out.
the first biggest branching gorgonian.
What's the prize?
I don't know.
Guess I'm going to recalibrate this right here.
Sure thing, thank you.
I'm just
I don't think
not
it could be
convinced.
anything food related because there's so much.
First cut at the potatoes.
Yeah, French fries.
Well, we're at pretty low oxygen right now at 3.4 micromoles per liter. Paul, compared to where we've seen the last octocorals or gorgonians on the previous dives, where would you expect? Could we be coming up to some areas as we're going to be kind of going along flat and then going up another steep embankment?
Yeah, look at that.
Yeah, good question. my sense is that where our best chances of finding them are if we can find some outcroppings that are exposed to more current. And so I don't know what you think, Greg, but that seems to be the most important variable that I've noticed so far in terms of where we might find some of these octocorals.
Off by almost 90.
Yeah, Paul, I agree. Once we're back up into rising oxygen, even if it's still low, we seem to do find the corals if there's good flow. And in about 100 meters, we'll be hitting a new wall, and I suspect we will see a lot on the slope going up, or I'm not guaranteeing it. You can never really predict what's happening. It's so variable down here, but it's likely we'll get some more corals on this slope going up. That's been our lucky kind of depth at about 600 to 500 meters with rising oxygen. Particularly if you remember a few days ago on the San Juan seamount, it was really a spectacular amount of corals. But also when we were on, in fact, I think it was this pattern escarpment in another area, we saw quite a lot of corals too as we came up through the oxygen minimum zone.
Beautiful little scene here.
Yeah.
There's a thorny head rockfish.
Beautiful.
Yeah.
Gorgeous fluorescent yellow sponge.
Interesting.
Brisingid.
Yep.
Look at that.
All sponges, apart from the
Yeah.
thorny head and the brisingid.
So we're getting some great fun jokes in the chat thanks to Desiree Zook. So I'll go ahead and and tell the first one.
Time for a joke.
Time for a joke.
Go for it.
Please do.
How do you amplify a pirate's DNA?
Something with R.
Arginine.
PCR.
Yes.
Yeah.
Oh.
Got it.
PCR. Oh my God.
That is wonderful.
A
I
really
like
good one.
that.
Yeah.
A good one.
It's been a while.
Fits perfectly with some of the molecular techniques Paul was just talking about. We'll use PCR a lot in trying to amplify the types of bacteria or specific microbes that we're interested in. That's a perfect joke, Desiree. Thanks for that.
Some impressive sponges coming up ahead at 50 centimeters tall. Two different kinds of glass sponges, one with a dirty sedimented outside and one that's immaculately clean, but both of them are glass sponges, just different kinds.
These are gorgeous.
Is that a different type of sea star, Greg, that we're seeing? That orange
Yeah, I
one?
see the one. It's not Rathbunasta, it's something else.
Did you want to look at it?
We can have a zoom, please.
Sure thing. I'll get us back into position here. All right, Jonathan, go ahead.
It's more than 5 arms, but it's not Rathbunasta. I'm not really sure what that is. Well, we haven't collected many sea stars on this trip. How about we grab that one?
You want that one? Raj. Full wide. I'm assuming this is going to be put in the forward box.
Where would you like this, Greg? Yeah. I've got the 2 forward bio boxes open.
And one is for various
Great.
miscellanea. So yeah,
Yeah.
forward.
Either one of
either
those.
side. Please.
Raj. never collected a sea star before, so this will be cool. Actually, sorry, I suppose. All right, Jonathan, go ahead and push in there tight, please. Ah, haha, the trick.
Right in the middle there.
There you go. Go ahead and
Yeah.
see if you can get a little more in there.
Ooh.
So it looks fuzzy. That's the sea star's got tube feet, little hydraulic appendages all coming out over its body. Echinoderms like the sea star have a water-filled system as well as a separate circulatory system, and the water-filled hydraulic system is how they move.
Come full wide there, please. It's probably be pretty stuck onto the rock, I would assume.
It's probably holding on, but it's using these tube feet to stick on, but it shouldn't resist too much.
Roger that. Push in there a bit, please, Jonathan. That's great, thank you.
Smaller than I expected.
Looks like it's getting larger.
Yeah.
It's pumping up to try and look bigger, scare you away.
He's got one arm up
Looking
going like,
scarier. Oh.
All right, slowly gonna close the jaws.
Little shrimp above it.
bonus shrimp.
Taking its home away.
Off it goes.
Geronimo.
All right,
Nice job.
Nice.
full wide, please. All right, Jake, go ahead.
Coming out.
Pop out the toolbox.
Desiree is saying it looks like a Solaster Delsani.
Oh no. All right, which way are we going? Left or right?
Either your choice, whatever's easiest.
Dealer's choice.
Oh no, little guy.
Oopsie.
In the box. In the box.
Sorry, little guy.
It wants to ride on the porch. Porch starfish.
Yeah. Let's try again. Don't want to squeeze too hard. Jonathan, push in there a bit, please. That's great, thank you. Jake, would you mind looking at it with the bubble, please?
Yes.
Thank you. All right, let's try that out. Full wide. Full wide. All right, I'll put him in the white one here.
Perfect. So this is going to be sample NA124 133, and it's going in the forward bio box number 6 white. And in it sink. Awesome.
Great. Thank you very much.
Sure. I wanted to let go before it squeezed it too much.
I know.
I was like, I see you're bulging out there.
Can I have that?
Oh, sorry, I was on SPL.
Thanks very much, Crispin and Desiree, for the name suggestions. We'll see if it's a Solaster. Not convinced, but we'll check it out. Our go-to expert is Chris Ma, world expert on sea stars. And I'll send a picture to him. He'll probably give us a name straight away.
All right, anything else around here that looks interesting to you guys?
No, I think we're good to go. Thank you.
Raj, come on up.
Bridge nav. .50 meters bearing 265.
Go ahead and push past the push cores there, please, Jonathan. Thank you.
Greg, we've got a question from you coming in from the science chat from Crispin Little, who's asking, what's the plan for the fossil whale bones and associated rock chips?
Yeah, good question, Crispin. I've stowed them away, took some more photos. It's definitely bone, it's no doubt. And I'll be back at Scripps on Friday and start to make a plan. I'll contact the person you recommended, and we have some paleoceanographers at Scripps, Dick Norris, I'm thinking, who works on forams, and ask him for some advice, and I'll probably reach out to you again once I'm home as well. There was no obvious evidence of Osidax in the bones. Some small holes, but nothing really clear. And so we're kind of in a little, not even sure it was whale. Right now I'm thinking marine mammal is about as far as I'd commit to on what it was, because it was only a few meters long. So yeah, we'll do a bit more work on trying to figure out what it really was, and certainly visually spectacular sight. If you didn't see it yesterday, go back to yesterday's dive and have a look at the video of this amazing black bones coming out of a white matrix.
So maybe dolphin, you're thinking?
It could have been a small beaked whale. It could have been a large elephant, an elephant seal. I think with those ribs, I think you're right. It's probably I'm not enough of a vertebrate anatomist to predict what it really would be.
Yeah, we did see a lot of dolphins.
I don't want to narrow down to whale right away, though. But we got quite a few chips of rock. Crispin, nothing obvious under the microscope in terms of inclusion, so we'll have to look at that more carefully.
The crab walking over there in the distance.
Oh, yeah.
Oh, yeah, across the rock.
Hands are up and everything.
Yeah.
Desiree Zook also asked in the chat if polychaetes normally are the ones with bristles along their segments.
Go ahead and push on there, Jonathan.
And yes, you were right, Desiree. Polychaetes do have bristles
Great.
along their segments. And actually their name means many bristles. Poly means many and chaete are the bristles.
Those are those
Go ahead, come.
carnivorous sponge, stalk sponge. I'm not mistaken.
Couple of those.
What's that little pink thing in the foreground?
Oh,
Is that a really small?
Is that a really small coral?
Yeah, Swiftia or?
I think they're Asbestopluma. That's a sponge, a carnivorous sponge.
Yeah.
Go ahead and push on in there, Jonathan.
And that
That is a little coral.
bubblegum, maybe,
Not really sure.
with a brittle star.
Oh
Tiny.
yeah, with a brittle star wrapped around it.
Yeah.
Yeah, that's the carnivorous sponge above.
Mm-hmm.
All right, full wide, please. I think that means Rennie wins the game, right?
No, only if it's good enough to collect.
Right.
I spy a hagfish.
Nice.
Always so cool to see those.
All right, Jonathan, go ahead and push in past the push course, please. Thank you.
All right, I got another joke.
Oh yeah, keep them rolling.
What kind of echinoderms come out at night?
Starfish.
Yep, sea stars.
There you go. Thought there was going to be another trick to it. That's a good one.
What about the vampire squid? That also would come out only at night, presumably.
Oh yeah. Oh yeah, with the vampire.
Mm.
I think in reality, lots of echinoderms and different vertebrates come out at night.
Could do a partial there, Jonathan. Great.
Ooh, very cool.
Or just
Persingid.
Persingid.
Yeah. Feeding away. Really nice shot of that.
There's a sea pen or something on the left.
Yeah, they're more of the carnivorous sponges.
Just behind the carnivorous sponge is a red thing is what I'm looking at. A red brownish. Right on that rock is swaying around. See that?
Go ahead and push on in there a bit more there, Jonathan.
Is that a
I think it's a rock pen.
Oh,
rock pen? Yeah.
So, yeah, I bet my money on rock pen. Full wide, please.
I bet my money on you betting your money on rock pen.
A bit floaty right now.
Here comes our wall up in Herc sonar.
Thanks, Nick, for Crispin for
an Argus too.
suggesting Nick Hyanson at the Smithsonian. I will reach out to him.
10-meter jump there.
Yeah. Sure is a target.
Yeah.
Very straight.
Looks like we're kind of in a sediment field with some smaller rocks now. A little bit different from what we've been seeing.
What we're talking about up here is we're anticipating, and you can see in Hercules sonar that,
Oh yeah.
and also in Argus, that linear wall that we're
Wall coming up.
starting.
Yeah, when we get to the wall, let's plan on doing a transect.
Sure.
Up the wall.
Up the wall. Copy that.
And though we might stop if there's corals, but it'd be a good one to transect up.
Sure thing.
Roger that. Up the wall or along the wall there?
Are we going up and over on the track? I can't quite see it.
I can zoom out here. We're on the track. We're supposed to go up to AD, and then at the ridge, follow the ridge along. We could always drop down if it's interesting back along.
Yeah, so let's
between
go.
A and AD
Perfect.
and E.
Let's transect up, and
Roger that.
hopefully along will be the best option at the top.
Copy.
What is the length of that track up?
Sure.
Top to bottom, is it 100 meters or?
Between AD and AE?
No, from the base of the feature to the top.
Yeah, 125.
Okay.
From where we are to AD is 200 meters.
Okay.
This one looks like it's just a step. So we
Yeah.
can start to see over it in Argus. Or sorry, in Herc.
Yeah, it's like a rubble pile.
Yeah.
So are we gonna, have we started the transect?
Well,
No, not yet.
I'm just looking here. This looks like just a step, and then it'll be flat on top still,
Right.
until we get to this main wall feature, which is I think what you want to do the transect
Yeah,
over.
yeah, let's get to the main wall.
Yeah, so we can just keep going over this.
Roger that.
Bridge nav. Add one step, 50 meters, 265.
It's a pretty broken ridge here.
Yeah.
We have a question in the science chat.
Look at that. Look at that.
Oh, yeah.
Beautiful.
Full jelly.
Is that a deep staria?
No.
No.
We have a question in the science chat from Desiree. How can we tell that we're coming up to a steep slope? Is it the slope on the Herc depth meter? Maybe we could, there are some things we can see that perhaps the viewers can't see.
Yeah, so we have some bathymetry that we've collected from the multi-beam sonar from the ship's hull. But when we're on the ground, we have our scanning sonars. There's one on top of Hercules, and there's one on the bottom of Argus. So we can kind of get a sense of what's coming up and before we can see it, we can anticipate steeper slope coming into view or flatter slope, whatnot.
And if she'd like, I'll actually just put sonar up on our satellite 3 feed.
Nice.
She can see, I believe, Hercules on the left-hand side and Argus is on the right.
That's correct. Yep. And those red rings on Hercules are 10-meter divisions, and on Argus are 20-meter divisions. So you'll notice that there's a dark space to the whole bottom left quadrant is dark on Hercules, and that has to do with where the scanning sonar is on Hercules. So Jonathan, if you do a quick zoom on Argus cam, you can see Hercules there. There's two black instruments on the top there, and the one on the towards us, which is on the starboard side or the right side of the vehicle, is the scanning the meso sonar that we mentioned. And so a lot of the vehicle is kind of occluding that bottom left corner. And the other slot is the USBL beacon for positioning.
those questions and jokes coming.
Do you mind recentering there, Rennie?
Sure thing. Sorry about that.
So we have
good.
another
Sorry.
science chat from Mary Wixton, who is alerting us that we should keep our eyes peeled for a deep sea hermit crab. Some of these deep sea hermit crabs have sea anemones or zooanthidians related to sea anemones on their shells. And so we wouldn't want to collect tiny ones because people really can't identify them from their photos. But it would be great if we could also be on the lookout for a crab the size of a tennis ball or bigger and get a frontal view. That would be really interesting, I think, adding to some of the biology that Mary is interested in.
Sounds cool.
Yeah.
Well, look out for sure.
Tennis ball crabs. Got it.
Have we seen any hermit crabs? I feel like we've seen some snails.
No,
A few on the last cruise in the Channel Islands this last week, but I don't think we've seen any this cruise, have we?
I thought I noticed, no.
Saw a couple that were in scaphopod shells, which was interesting.
I haven't seen many shells in general so far.
I think we could probably start a transect here and have the bulk of it be on the steeper slope.
The steeper slope. Beautiful. This will be transect number 3.
Copy that.
Has to be a 265.
265.
Raj.
Don't know if it'll be a wall, but it will be steeper.
Roger that.
You can start when ready.
All right, I am ready for you.
Okay, start of transect.
Beautiful.
Raj.
4, you said?
Oh, beautiful. Thank you.
Yeah.
Recording the time.
What? It was 3 or 4, sorry. I missed
Oh, transect number 3.
3, yeah.
Kira, yes, we did collect one rock just at the start of the dive. That would be R number 4.
it was a great rock. We love this rock. We hope you'll love it too.
We had a chat in the science chat from Desiree, who was asking if that was a red shrimp that darted on the screen. It was really too fast. I think we kind of see some of those on and off sometimes. They kind of dart across our screen. So yes, Desiree. And then Crispin Little's also chiming in a little bit about our conversation about looking for some hermit crabs, noting that there's been a paucity of gastropods in the rocky areas on this cruise. This is very different to the last cruise where there were bathybembics and euconids. I have a hard time pronouncing these all over the place on the soft sediment. So it's definitely a contrast that Crispin has been seeing between the different dives.
Oh, it looks like you have one near coral. Is that a coral to the left there?
Ooh, yeah.
Oh yeah, that's the little tiny white one with all the brittle stars that I can never remember the name of. Good sign for the start of the slope.
Yeah. Another rock pen there.
Mm.
Couple of short sign thorny head fish. Kind of hiding between the rocks. Looks like we've got that pink mushroom coral on the left side that we're going to be going over.
Mm-hmm. We should think about another collection of
Another nice coral to the right.
There we go.
Yeah.
Still
Precious coral?
Still pretty small.
Yeah, it's small.
That's true. I think we forget how nice a camera, what the true size is.
Yeah, we've just crossed 600 meters. We'll be coming up about a reasonably steep slope, and then next 100 meters or so. So we might see a bit of change in the fauna on the rocks as we go up.
Do we have an estimate of our rate of ascent here?
I'm holding about 0.3 knots over ground. So roughly about 0.3.
Cool.
So what would you estimate the angle of this incline is?
Judging on the bathy, it was about 20%. Sorry, 20 degree slope.
Yeah, that looked interesting. 20 degree, yeah, okay. Thank you.
Can be a bit difficult to tell visually.
Ooh.
Yeah,
This looks cool.
Is that a precious
Cool.
coral?
I think it's those, Steve's rolling in his grave downstairs because he's always teaching me all the different coral names. And, oh, there's a bubble gum.
Bubble gum.
Right.
Do you want that, Paul?
Paragorgia.
I
Wow.
wouldn't
Our
mind another one at some
favorite.
point. We have two already. Also wouldn't
Aw.
mind another one of the mushroom corals at some point, but
Well,
I'll mark a target there in case we need to come back.
yeah,
Or do you want to stop?
no. We can
Okay.
go.
Raj. It's probable we'll probably see another one as we get further upslope.
Yeah.
If they're starting here to be more abundant.
Yeah.
Yeah.
That'll be interesting.
They really do look like bubble gum, and they have really
Sure.
similar texture too, kind of
Yeah.
gummy.
Yeah.
Yeah.
Smells like bubble gum too. Just
Really?
kidding.
Tastes like
But does it?
Yeah.
Kylie would know.
Yeah.
Like
Anyone,
it's just like a gun.
Gotta use all the senses.
we get it. You eat sea creatures.
Paul, we've got a question for you in the sign chat that maybe you could lend some wisdom on. So Abigail Powell's noticing some of the bright yellow star eucalyptus sponges earlier, and they're wondering, it'd be interesting to know if the yellow comes out, if we do chemical analyses on it, would we expect the yellow to run out of it or be part of the tissue?
I'm sure it's an extractable pigment.
Oh, here's a good example of one.
Yeah, and we've collected this sponge and we are going to extract it. We'll extract it with organic solvents and we'll find out if whatever those pigments are organic soluble. If they're not, we tend to lose interest in them because organic chemists don't like to work on molecules that aren't soluble in organic solvents. But if it is organic soluble, then yeah, it'll be interesting to see what that pigment is. And yeah, quite a contrast, right? Because most of the sponges that we've seen have all been white.
Bubblegum coral, pink, little pink. But definitely a big contrast to that bright yellow.
I think that bubblegum corals are one of my new favorite animals.
We're starting to see a lot of the red brittle star that we noticed coming up San Juan a few days ago, and they became very abundant as we went through a particular area of oxygen. So this is, and shortly after we saw those brittle stars, we saw a lot of corals. So this is looking promising for more corals ahead.
Have you noted if there's been much oxygen change?
It's slowly rising. It's certainly out of the very low oxygen level. We never really got down to zero. But in the last hour, it's risen by 25%, by 33%. So it's steadily increasing.
Another big fan of a coral to the left, almost out of frame.
We're seeing some Panitia, sea cucumbers. There's cousin It again.
Not as spectacular as the one yesterday, but
yeah,
very similar.
Those spicules were crazy long.
Oh my gosh, yeah.
Okay, I gotta
Look at all the brittle stars. This is going to be a fun transect to count all the brittle stars. Wow.
And scare Devin away from that project.
Wow, lots of brittle stars.
We might be seeing that transition that Greg had just mentioned. In the science chat, Paul, we've got Abigail Meredith saying that yellow color of the sponges does come out in seawater and even just with alcohol too.
Yeah, good to know. Thank you.
And Meredith is...
And the sclerites, yeah.
Yeah.
So the sclerites themselves can be pigmented.
That's very cool. Some of these brittle stars are waving hello.
hello, goodbye.
There's a Dover sole hiding under the rock.
Yeah.
Just out of frame. Well, I think it was a Dover sole.
Looked like it. Pretty well camouflaged to the right there. We have a question from Desiree who's asking, how could we tell between a sea cucumber and a hagfish?
Oh, you can tell.
These are all sea cucumbers that we're seeing right now. Sea cucumbers usually don't move as much as hagfish. Hagfish are a little bit more gray.
Sometimes curled up.
And they have a distinctive tail. If we see the tail, it's pretty immediately obvious it's a hagfish.
That's a good question, though, because they are similar in size and shape.
Yeah.
Like,
Chris,
oh, sorry. Like sea cucumbers are a bit spiky too.
it's true. It's
Yeah.
true.
And the sea cucumbers never curl up the way a hagfish does.
Yeah. You've got Chris from Little also saying that hagfish wiggle a little bit more, and they have white eyes.
They have white something where their eyes should be. Just suck your soul out.
Oh, wow.
Not
Granny
that
is not a fan.
not that I dislike hagfish, right? Nightmare fish. Okay, that's the end of transect there.
Thank you.
Roger that. So we'll keep our eyes out for a bubblegum coral, a mushroom coral, is that correct?
Yeah.
Yeah.
And
Tennis ball hermit crabs.
tennis ball hermit crabs, Raj.
There's some mushrooms down there.
There's several on top of that rock just there that you're swinging to look at.
What was that? Start that?
Do you want me
Go ahead
to
and
hold up
push
the
in
ship?
there.
Oh, there's two different things. Yeah, there's
Oh yeah.
definitely Tetrapolypus on
snap.
top.
Oh, are those multiple?
Pull wide there, please.
Let's hold position. Thank you.
I'll get a better view for you guys.
There's a corallimorph, I think.
Oh, the